Yeah. We'll talk about DNA replication. DNA replication is the process of copying genetic information, genetic material for cell division. Like we have discussed about what is DNA in previous class and where it occurs and where it is located, what is the structure of the DNA. We have discussed. Then how the DNA will replicate means how, for example, there is a one cell, there will be formation of two cells, daughter cells, keep on dividing and every cell having the DNA. But how this many number of copies of the DNA which present in each and every cell and that too passes to the next generation. How it actually occurs. This is the process by which the DNA replication process through which the DNA will occur in each and every cell of an organism and it passes through next generation. Means by the definition, the DNA replication is the process of copying the genetic material. Just copying the genetic material. But in this class, we are going to discuss how exactly this mechanism will happen, how exactly the DNA replication will happen, that to which high fidelity, and it's very perfect without any mistakes. How much very stringent procedure. Still, we can't make any procedure as stringent as DNA replication till now today. Even though we have PCR, but it's not that much stringent as DNA replication. How exactly cell makes it happen? That we will going to discuss today. And this is the actual definition of just a copying a genetic material. Then another question comes, where actually it occurs? It occurs in S phase of the cell cycle. S phase of the cell cycle. It is in eukaryotes, but it occurs in the binary fission of prokaryotes. Then there are three different models for DNA replication. One is semi-conservative model, conservative model, and dispersive model. And dispersive model is nothing but entire DNA is fragmented, and there will be again this fragmented DNA will form a new DNA. Means complete fragmentation. There will be rearrangement of this one, multiplication of this one, again rearrangement of these fragmented particles and formation of the two different DNAs. In the conservative, entire new DNA is produced and both the strands are new strands. Together they will make and create a new DNA. But in semi-conservative model, in the daughter strand, one DNA is from parental origin and another one is from the new strand. It is called semi-conservative. How actually in normal the DNA it is actually replicated through semi-conservative model. How it is proved? How he disproved? How we disproved conservative and dispersive model, and how we accepted semi-conservative model? It is because of the great scientist Meselson and Stahl experiment. He actually conducted the experiments. With that experiment, he proved that the DNA replication occurs through semi-conservative model. Then, how it actually done? For example, he grown the DNA in N15 media in E. coli for the 15 to 20 generations. 15 to 20 generations. Then all the DNA which is having in the E. coli cells when it is grown in the N15 media, all are N15, N15. You to see that both the strands having N15, N15. When it is centrifuged in a cesium chloride and ultracentrifugation, cesium chloride density medium and in through ultracentrifugation, we'll get a band like this. Means all are, see the band is shifted to this is N15, N14, 15, N14, 14. See all band near to N15, 15 because only one type of DNA was present, that too having N15, N15 strand. When it is this DNA grown under N14 media for first generation, there is a hybrid molecule, one having N15 and one having N14. Means by this only we can get there is a DNA replication will not occurs to conservative. If it is occurs to conservative, what will happen? You are going to get either N14, N14 one band and N15, N15 one band. Means two band is to be present. But here only one DNA molecule were present, one type of DNA molecules were present that having one strand is N14, another strand is N15. Means here middle of the band is present. That's why he disapproved conservative model. Then, even after several generations, there is a slight band of N14, N15. Okay. But if it is a dispersive, if it is continuously grown in N14, then definitely there will be one kind of the DNA, N14 and N14 only. There is no presence of this hybrid. That's why he disproved dispersive. In dispersive, what will happen? There is all, if it is continuously grown in N14, N14, there is only, we expect N14, N14 bands only. But why N14 and N15 bands still present in the generations? Because of the DNA occurs in a DNA replication occurs in the semi-conservative model. That's why by these continuous experiments in three cycles in a different medias, he proved that DNA replication occurs in the semi-conservative model, and he disproved conservative model and the dispersive model. And why he used only cesium chloride as a density medium? Because the density of cesium chloride and density of the DNA are the same. That's why you use the cesium chloride. And it is done in E. coli. Please remember which organism he done the experiment, which medium he used, N15 and N14, and why he has used the density medium as a cesium chloride, and which experiment he conducted, how he disproved the conservative model, how he disproved the dispersive model, and how he, on what basis he told that it is a semi-conservative model. It's very, very important. Like, and in which year this experiment is conducted. Okay. Then it is in prokaryotes. But in eukaryotes, who conducted this experiment? Taylor and Woods and Hughes experiment in 1957. Actually, before only it came in eukaryotes. It is by use of H3H thymidine. By autoradiography experiment, he proved that the chromosome replication occurs in a semi-conservative model. That organism was Visia faber, broad bean plant. If you remember this much, is more than enough. Then how the replication will occur? There is a three steps in DNA replication was initiation, elongation, and termination. Three broad steps: initiation, elongation, termination. We will discuss initiation first. In initiation is actually the starting of the DNA replication process, which is the main sequence. You can see this one, where the initiation occurs means at the site of origin of replication. And what is consist of this region is consist of having 13-mer sequence and 9-mer sequence. Why it is so important? Where these 9-mer sequences are conserved sequences, where your DNA protein will bind, and it starts unwinding your DNA, double stranded. For example, your DNA is double stranded, starts unwinding when DNA binds. Then further it facilitates DNA B protein, which comes and bind in 13-mer, and it start DNA. DNA B is a helicase protein. When the DNA B will bind here, then there will be further unwinding of further unwinding of your DNA. Means DNA B. DNA B is the main helicase protein. First initiator DNA A, then DNA B, and DNA C actually facilitates the DNA binding to these sequences. Then again another protein called as SSB protein. After binding of DNA helicase and DNA A to this one, helicase will come and bind. What is the need of helicase? Whenever the DNA is double stranded, after helicase it will become single stranded. But again, there is a chance to rewinding. Like this, they will rewind and they will form the hairpin structures where you can't do a replication machineries will not move. To avoid this condition, your SSB protein will bind to DNA so that your DNA will be very much linearized, and it stops the re-annealing of the DNA structure, and it avoids the formation of hairpin loops. That's why DNA protein and B, C, and SSB are very, very, very important. Another protein called as DNA-G, where it acts as it adds 3' OH group to the group. It adds some will synthesize the primer, and it is makes availability of 3' OH, so that whenever the DNA polymerase will come and make utilization of this 3' OH, then starts your DNA replication. This is how the initiation will occur. First, DNA protein, which binds to 9 base pair repeats in ORC that we have discussed. Then DNA B is actually real helicase enzyme. It's further unwinding of the DNA. It separates the strands. The DNA C protein helps the DNA B protein. And there are SSB protein. It prevents the strand from reannealing. Then there is another enzyme called as DNA gyrase. When the DNA is supercoiled, like this, very supercoiled, no enzymes will bind here. It has to be revealed like uncoil. Like this, it has to uncoil. Who uncoils it? The DNA gyrase, which actually relaxes the supercoil structure to make all DNA proteins to come, DNA replication machineries to come to take the DNA replication. Okay. Then, normally there is, while DNA replication, there is an important one unique structure called as replication fork. There is a formation of replication fork like this, and this is like bidirectional in nature. This replication fork formation was actually identified by John Cairns in 1963 by autoradiography experiment. Then the replication occurs in both the direction, one direction or bidirectional, unidirectional or bidirectional. If unidirectional means the replication occurs in the both the sides. You can see here only one side, but here there will be two sides. In this condition also, you can see here in a very clear picture. Here, in this direction also DNA replication occurs. In this direction also, this is from origin of replication. In both the direction, you will get the replication. If it is bidirectional, if it is only one direction, then it is called as unidirectional replication. And please remember, DNA replication occurs through either unidirectional or bidirectional. But in many cases, the DNA replication is bidirectional in nature. Only in some cases, DNA replication is unidirectional. That we will going to discuss in coming slides. Then which are the main enzymes which are actually important for DNA replication is DNA polymerases. There are different types of polymerases. One type of polymerase is DNA polymerase 3. It is called as real replicase enzyme. Means why it is a real replicase enzyme? Because it's having high processivity. Means it will synthesize 1000 nucleotide per minute, I think per second or per minute. Please go through that. Means the efficiency of adding the nucleotides and formation of the new DNA is very, very high for the DNA polymerase 3 when compared to DNA polymerase 1 and DNA polymerase 2, where efficiency or processivity, the processivity is very important. Processivity is very low for DNA polymerase 1 and DNA polymerase 2, but they have higher processivity for DNA polymerase 3. And Othenberg, actually the scientist, like you just go through the which are the scientists when it is discovered, it's very important. Then when you look at what is the nature of this DNA polymerase enzymes? For example, you take DNA polymerase 1, the polymerase activity means the adding the nucleotide activity, polymerase activity, it actually in 5' to 3', 5' to 3', and in all the enzymes also it is 5' to 3'. But unique feature of DNA polymerase 1 is it having exonuclease activity, means removing the nucleotide also in 5' to 3' and 3' to 5'. It is the unique nature of DNA polymerase. Along with 5' to 3' polymerase activity, the same 5' to 3' activity, it also acts as exonuclease activity and also proofreading. That's why it's very, very important to know this unique feature of DNA polymerase. Normally, what is the function of this DNA polymerase is filling of the gap after removal of RNA preamble. That we will discuss, like while discussing elongation termination procedure, we will discuss how it will be helpful. Then DNA polymerase 2 helps in the DNA repair, and DNA polymerase 3 actually helps in the replication. and proofreading and editing. And the polymerase and the exonuclease activity is 3 prime to 5 prime. Normally, the polymerase activity 5 prime to 3 prime and exonuclease activity 3 prime to 5 prime. Exonuclease or proofreading activity 3 prime to 5 prime. But in DNA polymerase 1, the unique feature is in 5 prime to 3 prime also, we having the DNA replication at in 5 DNA replication at this one 5 prime to 3 prime direction. Exonuclease activity DNA at 5 prime to 3 prime direction. And please remember, the DNA polymerase 3 is having higher processivity than those other two organisms. And they're all monomeric units, but DNA polymerase is having multi-unit, multi-subunits like beta complex, theta regions, helicase region. All these regions are very, very important. It's a multimeric enzyme. It's having multiple polypeptides. So to hold the DNA not very tight, and it will enables high processivity. Okay, that you should remember. What is structure? It's not needed for SRF or ASRB. If you are reading for CSR, please read in detail about the structures and what is the function of each complex. Okay, what is beta complex? What is chimeric complex, processivity complex, alpha complex? Everything is very important for CSR. But as of just remember what are the DNA types of the DNA polymerase and what is ORIC sequence, what does it consist? Either DNA replication occurs at what direction? Normally it is bidirectional and unidirectional replication also occur in some of the conditions. And what is the main important proteins that involved in the initiation of the DNA replication and in which model the DNA replication occurs, it's very, very important. Then when it comes to initiation, first as I told, where your DNA helicase will bind, then it unwinds the DNA. Unwinds the DNA, then SSP protein will bind, then slowly there will be primer synthesis will happen by DNA G. Then this DNA will provide the primer and provide the 3' OH free group. Then slowly the DNA the nucleotide started adding. Then your chain will grow like this. Then how elongation will take place? Means elongation normally DNA replication always you remember it is in 5 prime to 3 prime direction only. Always your DNA replication occurs in 5 prime to 3 prime direction only. Okay. Then which is the main enzyme? DNA polymerase 3 enzyme which adds the nucleotide continuously through the growing chain. Then in this process there is two strand. One is lagging strand and another one is lagging strand, another one is leading strand. For example, in easier purpose, one is lagging strand, one is leading strand. One is lagging and one is leading strand. Means in a leading strand where DNA replication occurs continuously. See, here DNA replication occurs continuously. It means it's a leading strand. Lagging strands where DNA synthesis occurs, it's not discontinuously. Means there is a formation of Okazaki fragments. Means Okazaki fragments. Means short sequences will form. Further, these are joined by some enzymes called as DNA ligase. If the the replication of the strand will occur through the formation of Okazaki fragments, then after that, Okazaki fragments are joined by DNA ligase. That replication is called as lagging strand replication. If the DNA replication start continuously without any formation of Okazaki fragments, that is called as leading strand. And it will occur in both the direction. This is origin of replication. It will occur through here and it occurs through here also. This is called, and remember, lagging strand, what is leading strand, and replication occurs both bidirectionally and unidirectionally. And which are the proteins are very important to make this one possible. Then there are DNA primase which actually produces DNA G, it produces RNA primers. We'll see. Produces RNA primers. Then where 3' OH will be there, then there will be synthesis of this one DNA. Like parental DNA, there is a formation of these all molecules, SSB proteins. Then DNA polymerase will come, then start synthesizing the DNA, then it will be formation of this one. See, the Wakajaki fragments were discontinuous. Where is the leading strand? There will be continuous replication. And always replication. Why this leading and lagging strand will come? Because DNA replication occurs in only one direction, 5' to 3'. But opposite direction, definitely it having the discontinuous only. That's why you should remember how it occurs and what are the proteins are very, very important. Then it elongation means simply after primase adds 3' OH, continuously adding the nucleotide to it, and there is a formation of phosphodiester bonds, then your lead chain will occur. But somewhere it has to stop. Your chain is grown, but somewhere it has to stop. How it occurs? In prokaryotes, E. coli replication ends at TER sequences. And TER proteins will come and bind to these TER sequences and replication fork will stop. But in eukaryote, it's a very problematic because here we have required a special type of enzyme called as telomerase enzyme. It's very, very important. Telomerase enzyme. Like in eukaryotes, there is a problem called as end replication problem. End replication problem that actually solved by this enzyme, telomerase enzyme. That how it occurs, we will discuss in coming slide. Like before that, discussing the termination of this one, first we have to discuss how the replication occurs in the eukaryotes. In summary, how the replication in prokaryotes, first DNA B protein will come, unwinds the double helix DNA, then SSB protein will prevent the DNA strand from reannealing, then topoisomerases like gyrase 2 will release the supercoiling, then primase DNAG, which synthesize the short RNA primers, which provides the 3' OH, so that your DNA polymerase make use of this pre-3' OH, then adds the nucleotide continuously. Then DNA polymerase 3 enzyme, it actually main replication enzyme, which which adds the nucleotides. Then DNA polymerase 1, it's helpful in the removal of RNA primers. Like every RNA primers are synthesized, 3' H is released. But after replication, you have to remove this like RNA primers. Who will remove it? DNA polymerase 1 will remove it. Then in a lagging strand, there will be formation of Kazaki fragments. Who will join it? Actually, DNA ligase will join it. If you remember this much, is more than enough for the SRF exam or ASRB exam. Then how the DNA replication will occur in the eukaryotic organism? There are, in prokaryotes, the origin of replication only at one site. Many times in prokaryotes, there are some prokaryotes we having multiple origins, but in general, most of the prokaryotes having single origin of replication. That you should remember. Most of prokaryotes having single origin of replication. Single origin of replication. But in case of eukaryotes, we having multiple origin of sorry multiple origin of replication. Then that's why we can replicate the entire large genome in a very short time because of multiple origin of replication. Means what it means multiple origin replication? Many sites we have in one site. Means here also replication will start, here also replication will start, here also replication will start. But in the case of prokaryotes, we having single origin of replication, so that it occurs, the replication will occur at single origin. Then eukaryotes replication occurs at multiple origin. That is a unique, the striking difference between the replication of eukaryotes and the prokaryotes. Then here there are different types of enzymes we are used in the DNA replication of eukaryotes. One is polymerase alpha, polymerase T like theta, then polymerase epsilon, then polymerase gamma. Gamma polymerase actually mainly helpful in the like this symbol will come is gamma polymerase, which helps in the replication of mitochondrial DNA. Mitochondrial DNA. And alpha is actually like in DNA G, it acts like DNA G. In prokaryote, we having DNA G, it synthesizes the primer. But in the case of eukaryotes, we don't have DNA G. We have polymerase alpha, which provides primers. We're also having polymerase activity. Then another one is this theta having synthesis of lagging strand and epsilon is for leading strand. Please remember many times asked in the exam. Then one of the difference between prokaryotic replication and eukaryotic replication is the size of the Okazaki fragment. This question was asked in the 2023 exam. Size of the Okazaki fragments in eukaryotes or prokaryotes. In eukaryotes, we have smaller size of Okazaki fragments, 100 to 200 fragments. But in the case of prokaryotes, we have larger size of Okazaki fragment, 1000 to 2000. Normally, we confuse it. That's why remember this difference also. The size of Okazaki fragments is one of the striking difference between eukaryote and DNA, eukaryotic replication and prokaryotic replication. And termination also. Here, ter sites will be there, but Tusk protein will come and will block the replication event and replication will end. But in the eukaryotes, we're having a different problem called as end replication problem that can be solved by the enzyme called as telomerase enzyme. Okay, how this will happen, we will see in the next coming slide. Like as we said, it having multiple replicons like see origin, 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 where multiple sites where replication forks will be there. Then here another difference is, the DNA is packed in eukaryotes in the nucleosomes. But you should replicate your nucleosome. Then along with your DNA replication, you should also replicate your nucleosome. That is one of the difference. Then another one is telomere replication. See, we have a problem. We don't have primer to replicate this one. After removal of this one, we don't have and who will replicate this one? Here primer is there. We will remove the primer. But who will replicate this one? That problem is avoided by the one such enzyme is telomerase enzyme. Here primer is we will remove. But who will replicate this strand? That is was actually done by one enzyme called as telomerase enzyme, where it is a reverse transcriptase enzyme. It synthesizes the extended part of this DNA, and then again this provides as a sequence for to further replication, and DNA polymerase will come and again synthesize the new complementary string. Then this extension of this one happens, then replication will happen. For example, you take first telomerase will bind, then telomerase extends this 3' end like this. There is extension. See here it was like this. There will be extension of this end. Then this is by RNA templated DNA synthesis. Then this translocation of this one will happen, like this will translocate. Then the repeat extension and translocation several times. And see how much extra length is formed. Then after that, the completion of the complementary strand by DNA polymerase, where this DNA polymerase will synthesize the extended part like this. Then your DNA polymerase again will synthesize and fill this gap. Then again this extended part will remove. This is how the end replication problem is solved by telomerase enzyme. You should know what is the function of telomerase is actually reverse transcriptase enzyme, which is provides RNA templates, then synthesizes the DNA strand and extends the DNA strand. After extension, this will provide the base group. Then your DNA polymerase will fill this gap. Then your end replication will happen. So this is actually normally the actually occurs in cancerous cell. Normally, this problem is good for human health. If telomerase, high activity of telomerase ends, the cell will become immortal. It will not go for apoptosis. If the cell it's not go for apoptosis, what will happen? The cells will become cancerous cells. That's why this is one of the striking feature of your cancerous cell. Higher activity of telomerase enzyme is actually the nature of cancerous cells. Means it will become immortal. Normally, cell has to die when the time comes. Like apoptosis will occur, their natural death of the cells will occur, their new cells will generate and will it will be surviving. But if it is not occurs apoptosis, if telomere is continuously, means your end replication will not happens. What happens if end replication will not happens from one generation to next generation? There will be the DNA strands become shorter and shorter and shorter. Finally, the information will go into loss and cell will be died through apoptosis. But if this problem is solved, the cell become immortal. Then it will become a cancerous cell. That's why it's a very important in cancer biology. Telomerase action. Okay, for us it's not required. Who actually discovered this process? It's many times asked in the exam. Elizabeth Blackburn and Carol Greider actually discovered this end replication problem and role of telomerase enzyme in the solving this end replication problem in eukaryotes. Then how proofreading will occur? See, I told DNA replication is very stringent in nature. Normally, we expect very minute error. 10 to the power of 3, I think, minus 3. How much? Why this accuracy will come? Because of the proofreading activity of the DNA polymerase. Means 3' to 5' exonuclease activity will remove the incorrect bases. This ensures the fidelity and proof. This proofreading activity maintains the accuracy of the information and the fidelity of the replication. That's why this replication is very stringent in nature. It's a very less error. But this activity lost in the Taq polymerase enzyme. That's why PCR having more errors. Okay. Then DNA polymerase 1 has the having proofreading activity that is having in 5' to 3' direction also. That is a unique feature. Then along with this, there are several repair system, mismatch repair system, and there are several repair system like base excision repair, nucleated excision repair, recombination repair. That is not I'm not going to discuss. First, you should read the what is the repair mechanisms and how proofreading will happen, and which property of the polymerase enzyme is actually involved in the proofreading activity. These are very important. Okay. Then, how this DNA replication is regulated? There are several checkpoints. This is also many times asked in the exam. There is a one checkpoint called as G1 S phase control. Actually, if it is not replicated in S phase, the DNA replication was not takes place nicely. It will not go for further cycle. Mitosis will not happen. It checks the process here only. Only after proper replication only the DNA replication will the cell cycle will further continues. There are several cell cycle regulation mechanisms many times asked in this ASR exam, but it's not that much required for us. You just remember there are several checkpoints to regulate either proper DNA replication is happening in the cell or not. One such checkpoint is G1 space control checkpoint. It's very important. In eukaryote, germinin protein is very important in the re-replication, preventing the re-replication. Once it is replicated, again it will replicate means unnecessary duplication of the copies. To avoid that, germinated proteins play a very important role. You should remember this much is more than enough for the DNA replication. Then, in summary, where is the summary? DNA replication occurs in semi-conservative process we have discussed, and means each DNA molecule contains one old strand and one new strand. Then it having the originates in specific region like REC region. Then helicase will unwind, primase will add primers, and DNA polymerase synthesize the strands. And we discussed which are the DNA polymerases, which are the DNA primers, which are the different helicase proteins we have discussed. Then occurs in two strands. Leading strand is continuous and lagging strand is discontinuous replication. Why this? Because replication occurs in only 5' to 3' direction. The DNA polymerase proofreading and which one is leading strand? Normally 3 to 5 is leading strand and lagging strand is 5' to 3'. Okay, that you should remember. Then DNA polymerase having proofreading activity along with polymerase activity, and which domain having proofreading activity is 3' to 5'. Okay, that you should remember. Then eukaryotic replication is more complex than the prokaryotic because we having multiple origins and we have to replicate also nucleosome also, chromatin also. That's what's a very complex when compared to prokaryotic replication. Along with this, there are special type of replication. One is called as rolling circle replication and D-loop replication. In rolling circle DNA replication, you can see here it occurs in some viruses or some other organisms. Here it's a unidirectional, but your bacterial or eukaryotic, they're generally bidirectional in nature. But in some organism, this rolling circle DNA replication is unidirectional in nature. That you should remember. Then there is a, for example, you see here Nix will created. There will be synthesis of one strand, new strand will displaces the other strand. So that this displaced strand and new strand will form. Millions of copies will form. It occurs in plasmids, some viruses. Okay. This is called as rolling circle DNA replication. Then another type of called as D-loop replication. There is a formation of one loop within that your replication will happen. Okay. That having two strand like heavy strand and light strand. Okay, there is a formation of D loop, then strand synthesized in that loop, and there is a formation of new strand. Generally, this occurs in the mitochondria and chloroplast. And you should remember this much is more than enough. D loop replication and rolling circle DNA replication, and how the summary of how this replication happens, different proteins and different enzymes, and different steps: initiation, elongation, termination. How termination happens in the prokaryotes, how termination happens in eukaryotes. All these things are very, very important. This is about the replication. If you want, if you this is why I'm discussing very in a shorter way. For this is actually the exam is crop improvement. Our group is crop improvement. We should more focus on breeding aspects, genetics aspects, how genetics will help in the breeding, and PGR and seed. This is our domain. There is a separate domain called as MBB, molecular biology students. They had to read more about this. Like what is the you can take DNA replication for weeks. In normally we have one course like fundamentals of molecular biology. You know there is a syllabus is replication, transcription, translation, and gene regulation. The four topics, one course, fundamentals of molecular biology in MBB fine art. I think many of you covered. Here replication they thought for months together. Like at least 20, 16 to 20 class they will teach. There are n number of things in the DNA replication. Like if you read Pranav Kumar book, you will get in detail. If you want to read Pranav Kumar book Life Science Part 2 about DNA replication chapter. It is very nice. But for SRF, that much also not needed. To just read genetic made easy or nested and Simmons, I had to call these diagrams and information, everything from nested and Simmons. This much only there in the nested and Simmons, that much only enough for the exam. Okay. That's why more focus should be on that topics. What are the things I have discussed is more than enough for the your SRF exam or ASRB exam from this DNA replication chapter. If any doubts after watching this recording, if any doubts, if you are free to contact me, if any doubts you can ask and we can clarify your doubts. Okay. Bye bye.